can't hear me. That's right. We're going to start very, very shortly, so if you guys can come on in, and as usual, there's plenty of seats up front. You don't all have to sit in the back. And those lights are really bright. Can we turn the lights down a little bit? I look better in dimmer lighting. I'm going to try to start right on time because John has 75 slides and I don't want him to have to rush through them or to skip the slides at the end. So it's my pleasure to welcome John Yates to the stage for the ABRF Award for 2026. Now, John needs very little introduction. He was on the JBT editorial board for years. He's attended many of these meetings, but for those of you who are not familiar, currently he's the John Lyon Young Professor in the Department of Integrated Structural and Computational Biology at Scripps in La Jolla. He received his BA in zoology and his MS in chemistry from the University of Maine. He worked with Donald Hunt, which we're all familiar with. He did a postdoc with Leroy Hood at California Institute of Technology. He was a professor, associate professor with tenure before moving to Scripps. And I'm going to ask you, this is a procedural question, is at the end when we do questions, if you could please come to the mic so that basically not only we can all hear you, but we can hear the question that you asked so that people in the back also know the question that was being asked. And again, we're going to ask you to wait till the end for the questions. All right, without any further ado.
All right, thank you. And I want to thank the awards committee for bestowing upon me this great award. I learned last night that these awardees frequently, often, sometimes go on to win Nobel Prizes. And so last night I booked my air ticket to Stockholm. These are my disclosures, which is becoming more customary these days to put those up. So this is my view. Sometime in the year, driving home from Scripps, get stuck in traffic at the beach, and then the sun starts going down, and I get this great view of the sun. If you've been in places like that, you know that there's supposed to be a green dot when the sun goes below the horizon, and the marine layer often gets in the way, so we don't get to see the green. I don't get to see the green dot that often. But I love this title, "The Journey is the Reward," because it reminds us that life is short. We should enjoy it. We should have fun. We should go to conferences and have fun with people. And I am impressed that I found the people at ABRF to be incredibly friendly. We in the mass spec field are not as friendly to one another. But anyway, so what I thought I would talk to you about is some of the background of my journey through protein chemistry and so on and so forth. And I've been to about half of the ABRF conferences over the years, going back all the way to when it was at the Protein Society. But before I do that, what I'd like to talk about quickly is this issue that with all the chaos going on in the government and funding and so on and so forth, it's important to remind ourselves who really is the biggest driver of innovation in science. You know, is it the venture capitalists? Is it industry? Is it the pharmaceutical industry, academic researchers, government? I don't think it's the venture capitalists. They want to get their money back in 5 to 10 years, and they want to keep capital gains taxes low because they're big, risk takers and yada, yada, yada. Industries, some industries are pretty innovative. Pharmaceutical industry, maybe 50 years ago they were very innovative, but now they're big behemoths and they mostly just buy their innovation by buying up startups. which have mostly come out of academic research labs. And they spend about as much on internal research as they get back from tax break from the government. So basically, we're funding their research through tax breaks. And academic researchers love to say, hey, we're great innovators, and we are, but we do require funding in order to do that. And where does that funding come from? It comes from the government. And so I would argue that the government is really the big driver of innovation, because they fund things, and they fund things with the intestinal fortitude that a lot of industries are not willing to do. And this idea of government funding research really comes from Vannevar Bush. He was director of the Office of Scientific Research and Development during World War II. He funded a lot of the developments that were needed for the war effort and so on and so forth. His previous job to that was he was president of MIT. Interestingly, about half that money that was spent went to MIT. And then this picture on the side is one of the first NIH study sections, 1945, just after the end of the war. And so science, health research and so forth is starting to be funded by the US government as well. If you want to know what the topic of the RFA was, I'd be happy to tell you. It's probably not appropriate to say what the topic is here, but you can, it also is a sign of the times, all men, all white men, some in uniform, just fresh out of the military and so forth. So there's this great book that I read, Mariana Mazzucato. She's an economist, and she argues that the big risk taker is the government, and the government is willing to fund research for very long periods of time, longer periods of time than industry and the private sector will not do. And one of the things she goes through in this book is she talks about the iPhone and all the technologies in the iPhone and where they came from. A lot of them came from DARPA funding. And DARPA really drove a lot of the technology, and all Apple did is they assembled all this stuff into a nice little package, designed it really well, and so on and so forth. Not to say that that's not an easy thing to do, but really they didn't spend 30 years developing the GPS or voice-activated requests and so forth. So basically, the public sector socializes risks, so we pay for that through our taxes, and rewards are privatized. So the big investors like Peter Thiel make a ton of money off of Facebook, which is enabled primarily because of the internet. And so one of the great things that the government did was they funded the genome sequencing project. So it was less than the projected $3 billion, and it took a shorter period of time than was expected, but it was probably one of the milestones of modern biology, is that we have all these sequences now, and we just keep generating more and more sequences, because out of this came, out of the project also came new technologies for analyzing DNA sequences and so forth, and microarrays and RNA-seq, and all these great things that came out of that. But one of the things that was not anticipated was that this would be an important game changer for protein biochemistry. And if you look at the white papers and all that kind of stuff, nobody really talks about that. They talk mostly about bioinformatics. But mass spectrometry has also been a beneficiary, not to the same extent that DNA sequencing was, from government funding. And it really was a much tighter relationship between academic labs and industrial labs as they developed technologies along the way that became better and better and really improved things to allow us to get from just analyzing small molecules or atoms to be able to do entire proteomes now. The thing that drove me into, so I was a zoology major, I really liked biology, and then I took organic chemistry, and I loved organic chemistry. And I also got introduced to mass spectrometry through that. And one day they had a new assistant professor at the University of Maine when I was there, and he came out of Klaus Biemann's lab, and so he was doing GCMS to sequence peptides. And so I got really interested in that because it allowed me to combine zoology with chemistry and so on. And then this paper came out in 1981, came out from Don Hunt's lab, where he was using this new technique called fast atom bombardment. And if you're really old, you'll know what that is. If you're not, catch me later, I'll tell you. But it was a way to finally analyze underivatized peptides, get them into the gas phase so you could do something with them. And Don combined that technique with tandem mass spectrometry to sequence peptides. And in this paper, he only really looked at a tetramer, so met-arg-feed ALA, but showed he could sequence it, and that was going to be driving this idea that you could do protein sequencing by tandem mass spectrometry. And that eventually resulted in this paper, Protein Sequencing by Tandem Mass Spectrometry, published in PNAS in 1986. And really, everything that we still do today, the fundamental ideas behind what we do today, even though we're doing more protein identification, really came out of this paper. So it's a great paper. This picture on the, let's see if that's my right or your left, but the next picture, that's me and my partners in crime in graduate school, so Pat Griffin and then Jeff Shabanowitz. Jeff Shabanowitz was a senior scientist in the lab. He built mass spectrometers and kept graduate students in line, taught us things, and really was a great mentor in addition to Don. But the main problem with this method of protein sequencing by tandem mass spectrometry was simply that the interpretation of spectra was manual. And so I spent more time sitting on my desk trying to interpret spectra than I did actually running the mass spectrometer collecting spectra. And there's my trusty calculator that I had in grad school. It lasted, after 35 years, it was still running. 38 years, it finally died. I think it was either the solar panel or the battery inside died. But what you can see on my desk is a PC that I bought. Now I'm a graduate student, I'm making $8,500 a year. I spend $1,500 to buy a PC because I want to write software to interpret tandem mass spectra. And this was a state-of-the-art PC at the time, 8 megahertz, 8086 Intel chip, dual floppies, 64K RAM, real state-of-the-art. But fortunately for me, I wasn't spending my entire paycheck on this. I had gotten married to another graduate student who was also making $8,500. And so she probably, since we were in our honeymoon period, she said, okay, you can go ahead and buy that computer. But actually wound up paying back quite a bit. And so computers and mass spectrometry had been around since the 1960s. Really the mini computer started at MIT, and Klaus Biemann's lab started trying to integrate mass spectrometers and computers in various ways, from just doing calculations and also eventually collecting data and so on and so forth with the instrument. But people eventually started to think about using computer programs to interpret spectra, but it was usually just one spectrum at a time. And the programs weren't that great at being able to do this, but this is also what I was trying to do with my PC at the time. And writing programs when you got 64K worth of memory is not not all that easy to do. But I also got interested in this idea of databases. And so you guys probably remember Amos Baroque. He started the Swiss Prot, and he was also a past recipient of this award. And so they had taken all these mainframe programs that they had written for doing sequence analysis, and when the PCs started coming more common, they rewrote the programs and put them on a PC. And I convinced Don that we needed to buy that, so I bought it, and I had it in the lab, and it had a small database in it. Probably had 1,000 sequences in the database or something like that. But it allowed me to get in and look at all these sequences that had been around for a while. And this is, those of you who are my age will remember that IntelliGenetics was a private company that had the contract for GenBank. If you wanted to access GenBank, you had to go over Telnet. And so it was not easy to do, but they had this little contest, tell us how you use our software tools. And I came in third with my little write-up here on PC Gene. But in this little write-up was this idea of doing peptide mass mapping as a way to try to identify proteins. But it wasn't using MALDI time of flight, but it was using, because it didn't exist at the time, but it was using fast atom bombardment of proteins to try to find things that might be similar to the type of protein that we were looking at. And of course, also percolating alongside at this time is DNA sequencing, 1977. Sanger's paper comes out, Maxim Gilbert's paper come out, and everything is changing. Paradigm shifting, disruptive innovation, creative destruction, you name it. Everything's starting to change. And then, unfortunately, in 1978, Alan Malcolm wrote this paper, The Decline and Fall of Protein Chemistry. And I don't know if that was an editorial change or whether he intended to put that title in there. It was really kind of an unfortunate title, I think. But the paper was fundamentally right, that everything was going to change because now it's more straightforward and easier to sequence the gene than it is to sequence the protein. And so that was his main point. I think he got a lot of ridicule from the protein chemistry community because of his title there, but he was fundamentally right. And so after grad school, I went off to Caltech, to Lee Hood's lab. You'll see that's Lee at least on my right, all the way on the side. And Lee's lab was famous for doing a lot of different things. He created basically the modern core facility where he invented created the DNA synthesizer, the DNA sequencer, protein synthesizer, and then the protein sequencer. And he envisioned this idea of the microchemical facility where he would use this to do modern biology and things. And so here we are talking about this. At the time, Lee's lab was 70 people. And so he had the protein sequencing group, he had the DNA sequencing group, he had the immunology group, he had all these different groups going on. But most importantly, at that time, this is about 1988, 1989, there was this huge discussion going on in the lab. So Lee's lab had invented the 4-color DNA sequencer, and so it was kind of the epicenter of discussions about sequencing the human genome. This report came out in '88 from the National Academy of Sciences saying, yeah, we need to sequence the human genome. But everybody was thinking about or talking about how are we going to do this? We're going to do directed sequencing, we're going to do shotgun sequencing. How are we going to go about doing it? And they were also trying to make that 4-color sequencer actually work. I think it took a lot of effort on the part of a lot of academic labs in order to do that. Craig Venter's group was one of the groups that actually made it work it really well. But if you go through the National Academy of Sciences report, again, you don't find any references to protein biochemistry and how it's going to help. And so I'm sitting around at Caltech, and the discussions going on around the coffee pot are all DNA, sequencing the human genome, all that sort of stuff. And so I'm thinking, well, what role is protein biochemistry going to play? And more in particular, mass spectrometry in this era of genomics. And I had gone to Caltech because I wanted to take the 2D gels that they were using there and take the spots off and go into the mass spectrometer as a way to try to sequence them. And what they were doing at Caltech, Rudy Abersold in particular, was taking the spots off and putting them in the protein sequencer. And so we had always, in Don's lab, we had always thought that our fight was with the Edmund sequencer. We were trying to catch up with them and be more sensitive and all that. So this is kind of trying to figure out what I should do. And also in 1989 was another watershed moment in mass spectrometry where we got 2 new tools. We got electrospray ionization, we got MALDI. And so mass spectrometry was starting to become more and more friendly towards biopolymers, so to analyze any kind of biomolecule. We got this write-up in Science in 1989. They're talking about mass specs moving on protein sequencers and so on and so forth. And just to show you how snippy the mass spectrometry field is, there's this little quote in there, but its critics argue that no one but Hunt has been able to extract sequence from data from a quadrupole. There's no reference on who had that little quote. But you also see this quote a little further down where people are talking about how when you put electrospray and MALDI on magnetic sector mass spectrometers, magnetic sectors, they were the Orbi-trap of the day, high resolution, high mass accuracy instruments. The sky will be the limit. And the instrument they were referring to, unless you're in your 60s or something, you probably have no idea, it's this thing. And so you want to have that thing in your lab? The magnets alone weigh 2 tons. So turned out that both MALDI and electrospray never went on these instruments because they were incompatible with the way those instruments worked. And so they kind of went extinct as fast as the dinosaurs did 65 million years ago. So this is my little group. So I left Caltech and I went to the University of Washington, and this is my little independent research group starting up. And we're trying to sequence things. We get involved with the immunology group at the University of Washington, and we're sequencing some class 2 MHC peptides in the lab. And I have this aha moment because I had partially sequenced 4 to 6 residues. Then we would send it off to blast search. And we would do that because we wanted to make sure that we weren't sequencing some sequence that's already known. It's already in the database. And there was a lot of MHC sequences in the database. And so we'd wait for the BLAST server to come back, the email server to send us back the results. And I'm sitting there waiting, and I'm thinking, why not just search the sequence database with the spectra directly? Why am I doing this? So it's kind of my aha moment in 1993. And just to show you, or to illustrate issues with this idea, is that data access was not as easy as it is now. So we really couldn't get access to the MS data. They were in binary encrypted formats. This was part of the requirement from the FDA and so on and so forth, so you couldn't get in and change the data in the mass spectrum. And also sequence data wasn't all that easy to get. So remember that the World Wide Web really didn't launch until Netscape came out with their first browser, which was October 13th, 1994. That's version 0.9. And so in order to get sequence databases, we had to do FTP down to NCBI in order to pull out the sequences. Then the idea was how to compare spectra. So there were methods that allow us to compare spectra. So library searching was already a thing in mass spectrometry, had been since the 60s. But we would have to take sequences out of the database and then convert them somehow into a form where you can compare them against spectra. So how to reconstruct spectra from sequence. The data access problem, so we had limited access to MS data in 1993. There was this thing called NetCDF, and I thought that this looked like a manufacturer's slow walk just to appease the more vocal people in the field of mass spectrometry. But it could only output a single spectrum, so that wasn't particularly useful. Finnegan was a predecessor to Thermo Fisher, had given me a program to output an MS to a flat file format, but if we were doing LC-MS, it didn't really help. At the time, data-dependent acquisition didn't really exist anyway. And so what we did is I had hired a programmer by that point, and I had him, Jimmy Ying, crack the Finnegan data format, the binary file format. And we converted that to a DTA format. And then I called up Thermo, and I told them what I did, and they faxed me a non-disclosure agreement, and then gave me some software to be able to access it more formally. So they capitulated pretty quickly once we cracked their database, once I cracked their format. And of course, the genome projects had formally launched by about 1990, so protein sequence data was coming. We knew this. Some of the model organisms were being sequenced, so yeast was one of the first to be sequenced, E. coli. And so we could download databases like GenPep, and GenPep was just this sort of consolidated database where all the sequences that were being generated were stuffed in there, so they weren't divided by organisms, because that really wasn't a thing yet. But the Haemophilus influenza genome was published in 1995 by Ventnor's group. And so we had to deal with this idea of how to compare sequence to spectra, so we went through developed this process to do this. But more importantly, we had to figure out how to reconstruct, how to convert a sequence into a spectrum. And so we just arrived at this kind of strategy of making all the major sequence ions 50%, and then we put in the neutral losses that occur, and we put it in there. And this actually worked really well. And if you are familiar with this area of informatics, the mass spec informatics, people now have AI programs where they can predict with a great deal of accuracy the abundances of fragment ions that you might see based on a sequence. But I still contend that this method works better than that. And in fact, I haven't really seen that much of an improvement on our accuracy by using actual abundances. Where it does seem to help is when you're doing metaproteomics, and you've got these huge databases of huge sets of sequences of lots of different microorganisms. That seems to be one place where it does seem to help a bit. And so we had this other next issue was how to measure closeness of fit between the two things. And there was this nice paper by Kevin Owens. I give Kevin a lot of credit. He was using this cross-correlation analysis to compare mass spectral data, but he was just comparing, he was using it to do library searching. And so we used this technique, which required using fast Fourier transforms as a way to do comparisons, and it wound up being a very, very accurate strategy for doing these comparisons. And so it allows us to very accurately measure whether the sequence was a good fit for the spectrum. And we also, at this time, even though we didn't really have data-dependent acquisition, which is the automated acquisition of spectra, we did shotgun sequencing where we took yeast and we digested it up with trypsin, and then sat at the computer terminal and tried to very quickly collect tandem mass spectra as the peptides were flying by. And we showed that we could, in fact, identify genes or identify proteins based on using that kind of strategy. So this is the idea of no longer doing a gel electrophoresis, pulling bands off, but just taking the entire mixture of proteins, digesting and running it. So of course, as an assistant professor, I want to get a grant to do this. I also want to publish this paper. And so I submit a grant and gets a really bad score. I think it's at 73 percentile. And I really think there should be ways to review the reviewers, just like we do on Amazon, your product, because a reviewer can stand on Fifth Avenue and shoot your grant, and nobody's going to say anything. And so this guy, I know who he is, and he says, you're doing it all wrong, this is what you should do. And it turns out that he had developed this program for doing sequence alignment comparisons. And so he thought I should do de novo interpretation of the spectrum and then take that sequence and then use his program to do the search. It's an okay idea, except even to this day, de novo interpretation of spectra, even with our really great mass spectrometers that we have now, it's still not a very accurate method. Not even as accurate as the database searching is far more accurate than de novo interpretation. And so his idea, even 30 years later, still wouldn't cut it. So anyway, I would have given him a 1.0 on the Amazon scale for that. So then I try to publish this paper, and I sent it to PNAS, to Fred McLafferty. Fred McLafferty has a long history in doing library research, and so I figured he'd get this idea. And so they reviewed it, and he rejected it, but he gave me some really bad advice. He says, when you send it to the next journal, tell the editor, contact me, and I'll tell him who the reviewers were, and that'll speed up the process. And that turned out to be a really bad idea because the other reviewer really didn't like, I think one reviewer was Fred, and I know who the other reviewer was because he revealed himself to a friend of mine. And so I think that reviewer followed us around, and so I got rejected 2 more times. And this is where a mentor can really be an important thing in your life. And so I complaining to my former PhD advisor, Don Hunt, about this, and he says, do you have a copy of the paper? I said, yeah. And he takes it and he marches it over to the editor of JASMUS. That's the Journal of the American Society for Mass Spectrometry. And he says, Mike Gross, you got to take this paper. And so Mike goes back and he reads it. He emails me and says, oh, this isn't so bad. Send me the reviews and make the corrections and I'll take it. And so he took the paper, and this paper is the highest cited paper in the Journal of the American Society of Mass Spectrometry by a factor of 6. And whenever this is mentioned to Mike, he gets this big, huge grin on his face like the Cheshire Cat, because it was really helpful. And I learned a lesson from that as an editor of helping assistant professors with their papers. Maybe some senior person really ripped the paper to shreds, and I'll say, fix it, correct it. I'll get a third reviewer to come in and look at it and help them along. Because Mike was really important. This is a really important step in my career. So we very quickly went from here to doing searches of modified peptides, in particular phosphorylated peptides, and developed a process to do that. There are better processes now, but at the time, this worked really well. We also showed that we could take DNA databases, and we could convert everything into sequences, and we could use that, so what is now called proteogenomics. We did 6-frame conversions on that. And one place where we used this, where it wound up to be this idea of proteogenomics, is we were working on malaria. We were working on Plasmodium falciparum, Plasmodium yoeli, and this is an AT-rich genome. And almost all the software tools for identifying open reading frames were based on GC-rich genomes. And so this wound up being a really hard problem. And so we were working with people at Sanger the Sanger Center, that we're doing the sequencing, and we do all stop-to-stop ORFs we're creating, then we'd search our yoeli data against that, and that would help them identify particular genes that they were unsure about because their programs weren't all that well trained for AT-rich genomes. Okay, so by 1996, we have this ability to automatically interpret data. And so we're super excited by the possibilities. We've already demonstrated that we can take mixtures and we can digest them. So what we need are faster scanning mass spectrometers in 1995. Those of you who are familiar with this, the LCQ came out, which was an ion trap mass spectrometer, and it featured a lot more automation on it than did the previous triple quadrupole mass spectrometers. We're trying to make our searches go faster. And so we built a Beowulf cluster using parallel virtual machines. So we bought 12 DEC alpha computers and stitched them together. And this was fun. I mean, we were making fast computers out of this. And that's now, I mean, how everybody's doing everything now is by clusters. Data acquisition algorithms and data transfer got better because the LCQ, and we start working on larger scale chromatographic separation, start working on LC. I'd always been a big fan of liquid chromatography, and so this was actually a lot of fun too. And so we have this strategy that we developed for doing shotgun proteomics. And so we take protein mixtures, digest them, and then take the peptides and run them through the mass spectrometer. The mass spectrometer is getting more and more automated and faster. And then we can analyze the data. And more recently, a little company that spun out of my lab had developed this device called Pacer, which uses GPU searches. Bruker acquired the assets to that company, and this was a real-time search algorithm. So while the instrument's collecting data, you could be searching your data, and by the time the run is done, you're done with all your searches. But basically, we could collect all this data, output, and come up with a list of protein sequences. And so what I want to tell you about now is just some things that we're doing more recently. And so we've gotten into this concept of the 3D proteome, and so trying to understand the conformations, the structures of proteins, and not just in test tubes, but what they look like inside cells and inside tissues. And so we developed a method we call covalent protein painting. It's basically a protein footprinting approach. So we use formaldehyde. So if you do any proteomics, and you've done any quantitative proteomics, you'll recognize this method. There's a slight variation on it. So you use formaldehyde, creates a shift base, the lysine residues. We do that in intact cells. We try to keep proteins non-denatured. And then we treat it with cyanoborohydride. Cyanoborohydride converts it to a dimethyl tag. That can be done with light isotopes. You can use light formaldehyde, light cyanoborohydride. And then we split open the cells, digest the proteins using chymotrypsin, and then we relabel with heavy formaldehyde and heavy cyanoborohydride. And so now we have two different tags. And what this does is it labels the surface-accessible lysine residues. And then when you crack it open and digest, we then label the ones that are not surface-accessible. We can use the mass spectrometer to measure the changes that have occurred within the protein, quantitate that, and then we can tell what's solvent accessible versus solvent exclusion. And based on that, we can compare a control state to a disease state, for example. And we just recently had a paper come out in Nature Aging where we did this in 550 patient samples, controls, MCI, and AD, to be able to see if we could find some sort of conformational signature that was associated with AD, and we did. But what I want to quickly tell you about is just how we applied this to look at delta F508 cystic fibrosis, the cystic fibrosis ion transport regulator. So it's a protein that fails to mature. So we're trying to understand what's the mechanism behind this failure. So it's been known for a long time what the mutation was in the most common mutation associated with cystic fibrosis. But the drugs that were developed were all developed by phenotypic screening without having any kind of understanding of what the mechanism is. And so we started looking at protein-protein interactions and wild type versus delta 508. We looked at post-translational modifications. We found signatures that signal when the protein is mature. And then we started having questions about structure, and we started having these questions before the cryo-EM structure finally came out. And so we were using this footprinting method as a way to try to understand the structural changes that were occurring on the protein inside the cell as compared to the wild type. And though out of all this data, we've been able to develop a putative therapeutic that can rescue the protein. It uses a different mechanism than by which the Vertex drugs does. So the cryo-EM structures came out, took 2017. I always like to tell the story of when I first moved to San Diego in the year 2000. The Cystic Fibrosis Foundation gave a company that just started up in San Diego, Structural Genomics, $25 million to do the structure of CFTR. And it took cryo-EM and almost 20 years later for a structure to come out just for the wild type protein, which shows you how hard of a problem that really was, because it's not a very stable protein. And so it took another 5 years to get the Delta F508 CFTR cryo-EM done. And because they had to find, had to make changes to the sequences that stabilized the protein structure enough that they could get a really clear shot based on the cryo-EM. Okay, so we compared our CPP method to the cryo-EM structures. We have really good agreement between the 26 lysine residues that we can measure in the wild type versus Delta F508. But one of the things that we don't have to do is we don't have to remove sections of the protein in order to do these experiments. And so now we're looking at what the protein looks like inside the cell with all of its interactions and so forth. And we find some pretty big differences between at this K273. And so these blue dots down here are the wild type protein. The red dots up here are the Delta F508 protein. And so it's very inaccessible in the wild type protein, very accessible in the mutant form of the protein. So what is that? Turns out that it's in this ICL2, so it's intracellular loop 2 structure, which forms a ball and socket with the nucleotide binding domain 2 segment of the protein. And so the K273 gets buried inside NBD2 in the wild type protein, but in the mutant form of the protein is very exposed. And so that's telling us something about what might be going on, that it's uncoupled. And so we now take some of the Vertex drugs. This VX770 drug is an activator, potentiator, and so it causes the channel to open up, stay open. And so when we do that, this shows that K273 gets fully buried inside the NBD2 domain. So that's an activated, mature state of the protein. And now we look at wild type, so VX770 shows no accessibility. And then when we look at Delta 508, DMSO treated, it's up here. VX809, so that's another drug from Vertex that was supposed to be a chemical chaperone. We don't see that much of a change. The blue up here. And then Delta, and then the minus the 28 degrees C is a temperature rescue of the protein. It's a temperature sensitive mutation. And this is considered to be kind of the gold standard of rescue. So when you get the accessibility down to about here, the protein will still mature and get out to the cell surface. So this slide's always a mess because we haven't figured out how to present it any better. So it combines a whole bunch of different mutant states and treatments and so on and so forth. So another non-trafficking mutation is this N1303K. And when we look at this, it suffers from the similar problem. This K273 never engages with the NBD2 domain, showing that that's clearly an important component signaling that the protein is mature and is ready to exit. You can see that here, the N1303K, K273, it's still very accessible as opposed to what it might be in the wild type. So this allows us to develop a model for CFTR conformational changes. So when the protein's fully activated, K273 is buried. When we have partial K273 exposure, this is at the plasma membrane during activation, that's suggesting that it's mature. And then when K273 is fully exposed, this is suggesting that the protein is immature and will be targeted for destruction. So that's just a little vignette of what we've been able to do with this technology, combining it with other sorts of things to look at the conformations of proteins within the 3D proteome. So over this time period, we've had this convergence of technologies, 89 to 1994, so ionization, new ionization methods, electrospray, sequences from the genome sequencing projects. I am probably the biggest fan of the genome sequencing projects because I got a career out of it, and I didn't even have to sequence DNA. We got algorithms, we got new mass spectrometers, so once there was a firm direction for manufacturers to go and big markets at developing, they really pushed hard to develop mass spectrometers that would do proteomics in a much more effective and efficient way. And the mass spectrometers that are coming out now are just unbelievable, and both in terms of their performance and their price. So this whole thing was an unintended consequence of genomics, I believe. And I love this quote from Freeman Dyson: "New directions in science are launched by new tools much more often than by concepts." And I know I'm preaching to the congregation here, because that's your job, is to bring these new tools to the benefit of the departments in which you work, the universities in which you work. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained. And one of the great areas that I'm very fond of is this idea of doing paleobiology with proteomic technologies. I was on the science editorial board when the Tyrannosaurus Rex paper came in, and I told them, if this turns out to be real, this will be a huge paper, but it also wound up being a controversial paper. So this is these technologies have just really enabled all kinds of new directions in science. And so these important mentors, this is Don Hunt, Ian Jardine, who was early mentors at Thermo. He, probably when I showed him what we could do with the database searching algorithm, he was one of the few people in industry that got it. We approached every single manufacturer and offered them up the software, and he's the only one that jumped on it. And then Lee Hood, of course, envisioning large-scale biology, a bunch of people at Thermo over the years, a bunch of friends from the mass spectrometry field. And then I've had a lot of programmers come through the lab over the years, and then alumni. This current staff is down at the bottom, and then people who have come through the lab over the years. And then a bunch of people have come to the lab to do sabbaticals, including Mike Fitzgerald. I really credit Mike with having brought to my attention this idea of the 3D proteome. He developed this method called Sprock's mass spectrometry method. And he came to my lab on sabbatical because he wanted to take his Sprock's method, which was done in a test tube at the time, and then take it proteome-wide. Did that in yeast. And then for 25 years, I had been involved in this yeast resource center. This is a P41 center grant based at the University of Washington. We started it in 1997 as the yeast genome was coming to completion. And the idea was to use the yeast genome and all these different technologies as a dress rehearsal to try to understand what all the genes do. Stan Fields and I were the two original members that lasted to the end. There were a lot of people that came in and out. And you might recognize David Baker, the recent Nobel Prize winner for protein design. But back then, when David first joined, he was doing more protein structure analysis. Then he pivoted to protein design. And that's a more recent photo of lab and the alumni from who still are in San Diego. And then some of the early funding sources that have helped pay for this work. And again, my ride home. Thank you.
Hi, John. Sheena Mache, NYU. Great talk. It was, it brought back many memories, and it is wonderful to see the scope of your work.
Thank you.
So, congratulations on your award, and I don't have any questions. I'm just in awe of what you have accomplished. So,
You know, I tease some of my younger colleagues about how I feel bad for them because it's so hard to really distinguish yourself now. Because back in the early 1990s, literally everything we did was new and unique. So just an exciting time.
Kevin Knudsen, University of Iowa. I share Sheena's sentiments. It was like watching my career history with the ABRF and so many things that you participated in. Give us a nice history of where we're at today. Maybe you can predict a little bit of the future, AI and other tools, and how they're going to change our understanding of protein.
Yeah, I'm kind of hoping AI doesn't take my job. I'm really enthusiastic about 3 different areas. One is single-cell proteomics, but I think there are specific applications of that make sense. One of the things that we're doing is we're combining patch clamping electrophysiology with single-cell, looking at neurons, trying to look at specific circuits. We've just published a paper where we're doing this in live brain tissue. And we're using addiction as a model because my neighbor across the hall is an alcohol addiction expert. So I think that kind of makes sense. I think the cancer biology really makes sense because you want to look at the evolution of the cancer cells across, say, solid tumors and so forth. And then there might be some other ones that I'm missing, but those 2 really make a lot of sense to me in terms of single-cell proteomics. I'm also really hot on this idea of 3D proteomics. The other thing I didn't mention is that we're also finishing up, starting to write up a paper where we have found a biomarker for this disease, ATTR-CM. So if you watch TV at all, you've probably seen Morgan Freeman on TV talking about ATTR-CM. And one of my colleagues at Scripps had developed a drug for this disease, tafamidis, but there was never a diagnostic for it. Diagnosis was always very late in the process and done by imaging because people would start having cardiomyopathy. And so using this 3D proteomics, we found a marker for transthyretin falling apart. And it's fantastic, and it works really well. So I'm excited about that.
Hi there, I'm Elena Weinheimer from the Bigelow Lab of Ocean Sciences. I was very inspired, and especially hearing about coming up with new ideas and sometimes facing challenges with challenging the status quo and running up against like those editors. I was curious if you have any advice for early career scientists who have maybe unorthodox ideas or science that is struggling to break it into the mainstream ideas. And I know you mentioned having mentors that kind of advocate for you, but do you have any other advice for helping us get our ideas more accepted or heard when we're not exactly big names yet?
Yeah, so probably the most important thing is to be tenacious and believe in it. So at the time, the other thing that was going on is that with these new mass spectrometry techniques emerging is that 2D gel electrophoresis, that field was so excited because now they could finally identify those spots on the gel. And along comes this guy from the University of Washington saying, no, we don't need gels anymore. And so, and 2D gels were very popular in Europe at the time. And so there was one guy, Dennis Hochstrasser, big 2D gel person, who kept bringing me to electrophoresis conferences to talk. And what he was doing was, telling his colleagues, you guys better step up your game because this is coming for you. And so I really appreciated Dennis doing that because he didn't really have to do that. But with things like that, get out there, be tenacious. I wrote this editorial for JPR called The 12 Rules for Science, at least my version of it. And so some of the things are in there that I think are important lessons that I've learned over my career is, you run ideas by people, and sometimes they say, oh, that'll never work. Probably should try it anyway. Because I had the idea for data-dependent acquisition, and I asked an engineer at Finnegan at the time whether that would work, and he said, no, that'll never work. And so I delayed doing it and that somebody else did it first. So do the experiment. That's the motto of my lab.
Thank you.
Thank you for the talk. My name's Sean. I'm from University of Missouri, Kansas City. And so I run a proteomics core, and one of the things that we try to collaborate with our clinical research fellows, and they're always wanting to do diagnostics, trying to identify different diseases and develop new biomarkers and things like that. Do you see, because most of the time they just want to give us blood, and blood is not necessarily what's going to happen, what's going to have the proteins in it that are going to help diagnose. Do you see the future of proteomics to include this sort of medical diagnosis?
Oh yeah, absolutely. That's kind of my third area that I think is really hot right now, is that we're finally starting to make inroads into serum. And there are some of these new nanoparticle techniques, the SEER, and there's another one that's actually more cost-effective from series, where you can make these protein coronas as a way to do things. So that seems to be helping people a lot. I know the UK Biobank is going to have a call for proposals to run 50,000 blood samples. And so I know some of the mass spec labs that are gearing up to try to take that on. So yeah, I do think I think it's finally getting there. I had a lot of people early on in my career approach me about wanting to do serum, and I really stayed away from it because it just seemed like a real black hole to me, where the dynamic range, the abundance range of proteins was just not worth the time to try to figure out. Somebody was going to figure it out at some point, but probably going to take 10 more generations of mass spectrometers to get there. And I think we're getting there.
All right, we'll give John one more. On behalf of ABRF and the award committee, we'd like to give you this award.
Oh,
There you go. I know. All right, thank you. Oh, thanks, everyone.
thank you.
I have a name for you from the past. You know Charles Hauer?
Yes.
So Charles and I worked together on isolating the proteins in the kinetochore, and he said, make sure I say hi to you.
Is Charles still around?